Aloha. Welcome to lecture number 7. In this lecture, we're going to cover week 4, and more specifically, embryonic folding, which is super, super exciting, action-packed week. Let me get myself on the screen here. There we go. So basically at this stage of the game, we have got a flat disc. Okay, here's your flat disc, right? Your flat embryonic disc. This is going to be the cranial side over here and the caudal side over here. We've got this little blue circle in the middle of it. We're going to pretend that that is the neural tube. Okay, the neural tube, we're just going to imagine that that goes all the way through this disc, this rough, really should be maybe round, but this is going to be our embryonic disc just for purposes of this demonstration. So the embryo at this stage of the game, we've got all the germ layers formed. We've got the neural tube formed. And what we've basically got is a disc with a tube through it. And the problem with that is that the human body is basically a tube within a cylinder, or really a couple of tubes within a cylinder. So somehow or other, we've got to get from a flat disc with a tube through it to a cylinder with a tube through it, right? So the way that's going to happen is this embryonic folding. Now, what's going to happen is that these edges are going to fold down and under, and they're going to zip together. They're going to merge together, and that is essentially what's going to give us that tube within a cylinder kind of shape, if you can see that, right? We got that tube within a cylinder, but here's the problem, right? We've still got, this is the cardiogenic area, right? This is the heart, and then this is the oropharyngeal membrane, and then this is the cloacal membrane, which if you remember is destined to become the anus, and this is destined to become the mouth. They're all on the back, they're all on the dorsal surface at this point, right? So how are we going to get them where they're supposed to be? Well, the thing is also going to fold the ends in, right? So as these ends fold in and over, all of a sudden we end up with now the heart in the chest, the oropharyngeal membrane where the mouth is going to be, and the anus where the anus is going to be. Pretty crazy, right? And so you end up, and honestly, the real deal doesn't look so different from this. They're pretty ugly at this stage of the game. But yeah, so now we end up with the heart in the chest, the mouth at the mouth, the anus at the anus, and you've got kind of a real basic rough body format where you've got a tube within a cylinder, the heart's in the right place, the mouth and the anus are in the right place, and you can start to develop this thing into a more human-like form. So that is embryonic folding. At least that's my attempt at a visual aid for embryonic folding. Now, let's look at some pictures in the book that maybe explain how it goes a little bit better. All right, so let's take a look at this now. Let's get everybody oriented first to the picture, okay? So this guy here is looking at the embryo from the top, and this guy right here is looking at the embryo from the side. This is do you remember? Sagittal section. This is a transverse section. This would be a coronal section, but all I've done is cut off the amnion, and you're really just looking in at the top of the embryo. Okay, over here you're looking at the embryo from the side. It's more or less intact. You're looking at it from the side. And this one is again a sagittal. So all of these ones through the middle here are sagittal sections. All of these ones on the right are transverse sections. So you're looking basically, if you cut it in half and you're looking from the bottom into it, right? And then all these ones are just looking at the embryo itself. So this one's from, these ones are all from the side, and this one, of course, is from the top. All right. So it's important that you understand, first of all, that there is a big balloon of water around all of this, right? So they're only showing this, but there's a great big balloon of water all around this whole thing called the chorionic sac, right? You've got the amniotic sac that covers all of this, but then you've also got the chorionic sac that is a big balloon that all this lives inside of. Okay, so don't forget that. Now, the folding happens primarily as a result of one layer of tissue outgrowing the adjacent layers, right? All it is differential growth rates of all the different layers. That's what causes the folding. There's not some kind of crazy muscle contraction or tissue shrinkage or anything. All it is that one layer grows faster than the other layer. And the result of all of that differential growth is this folding process of the embryo, okay? It's also important that you understand that the lateral and the ventral folding, so the folding forward and the folding sideways, they happen at the same time. I know in my demonstration I showed the lateral folding first and then the ventral folding, but they're actually happening at exactly the same time. And that's kind of where we're at. Well, we're going to go over each one individually, but just remember, keep in mind that they happen at the same time. So let's go over ventral folding, okay? The ventral folding, like I mentioned, moves the heart and the pericardial coelom, right? So you remember there's that up here, there's this intraembryonic coelom, right? So this part of it that's up here in the cardiogenic area, they call that the pericardial coelom because, well, it's right underneath the heart, or right above the heart, rather. So as this thing folds forward, right, that pericardial coelom and the primordial heart tube comes with it, and it eventually winds up in the chest as it enlarges, right? And you see here on the lateral view, this is that great big swollen heart area, right? The heart is actually quite big compared to the rest of the embryo at this stage of the game, and it kind of needs to be. That's fine. But yeah, you have this great big heart swelling. Okay, and then this would be the forebrain here, right? So and the oropharyngeal membrane is right in here. So as this guy folds in this way, you get the heart into the chest area, the oropharyngeal membrane just above the heart, right? Now, as the back folds in, you have this cloacal membrane up here. Again, it's on the dorsal surface right now, but as this end folds around, this guy actually ends up, right underneath the tail, because embryos have tails still. And then what happens is, this would be the amniotic sac, right? All up around here, right? So the amniotic sac folds around as well. And as this happens, it looks like, so all of this basically pinches off. So this is allantois here. If you guys remember, allantois comes around and comes forward. This is the umbilical vesicle. So most of the umbilical vesicle gets pinched off. Allantois kind of gets pinched off, and this amniotic sac ends up surrounding the whole thing. So you end up with this big pinched off, and this is basically the umbilical cord. So the umbilical cord is made up of the umbilical vesicle, allantois, the connecting stalk, right? Oops. And that's what all of this is, right? See, this is the umbilical cord, basically, and that umbilical cord is made up of all of this stuff basically just getting pinched together at the belly. Because remember, the lateral folding is happening at the same time, right? So let's go over that a little bit, okay? So the lateral folds. So this section in A3 up here is taken right where the intraembryonic coelom opens into the chorionic coelom, right? The intraembryonic opens into the extraembryonic, the chorionic sac, basically. And so what's going to happen as this thing folds down, right, as these sides fold over, is that coelom is going to get significantly larger, right? So this is that intraembryonic coelom that we talked about, and it gets a lot bigger, and this is eventually going to become the thoracic and the abdominal cavities, right? Again, this is the umbilical membrane, right? So this is the lateral and intermediate mesoderm is what this part is right here. And you see how the ends are basically just folding in, folding together, and then this space right here gets a lot larger as it folds in, right? And so now you have this cylinder shape with a tube inside and a tube inside, right? This is going to become the gut tube. This is the neural tube, which of course is going to become the spinal cord, the central nervous system, and the spinal cord in the brain. And then you have the thoracic and abdominal cavities. It looks like where this section was taken is more like the upper abdominal cavity, or maybe even the lower thoracic cavity. This one's taken straight through the umbilical cord, right? This is the level of the section right here. Okay. And so this is what you end up with is, you know, the heart is here, the brain is here, the mouth is here, the anus is here, and you've got everything pinched together in the front into this umbilical cord right at the belly. Let me make sure that I didn't forget anything. Check my notes here real quick. Very good. Now, while all of this is happening, this amniotic cavity that we've been talking about, that, you know, would be right here, except it's been cut off in this picture. This amniotic cavity, as this embryo folds, gets bigger. It gets a lot bigger. In fact, it gets so big that it fills up that entire extraembryonic coelom. That chorionic sac becomes entirely filled up by the amniotic sac to where they're butted right up against each other. I don't know if any of you have ever seen a birth, but they talk about the membranes coming out, right? But yeah, basically there's two layers. There's the chorionic membrane and the amniotic membrane, and they're right up against each other. You have to work to get them apart after the baby's born. And this is when that happens. This amniotic membrane, this amniotic cavity expands and completely fills up the chorionic cavity. It fills all the available space, right? Interestingly enough, the amnion also winds up forming the epithelium of the umbilical cord. So as this all gets pinched off, right, this umbilical cord, the epithelium surrounding it, is formed from the amniotic membrane. This guy right here kind of folds around it. If you can picture that, it folds down around the umbilical cord and becomes the epithelium. Let's see. We're going to show you a quick video now. To give you an idea of, oh gosh, I skipped a couple slides there. Well, this is just a better view of how the oropharyngeal membrane folds down around into there. The heart folds down around into the chest. It's just a better view of what we already talked about. And then, you know, umbilical vesicle, connecting stalk, all that stuff getting pinched together into the umbilical cord. Sorry, I guess if I'd have showed you those two slides, that would have been a lot more clear. At any rate, let's see if this video works. This is a video that came with the book, or at least with the teacher version of the book. So you have, color-coded ectoderm, endoderm, mesoderm, and you've got the transverse view here. And this is at the level of the umbilical cord, right? So here we've got, let's back that up just a little bit to get you oriented. So you've got the amniotic sac up here. This is the neuroectoderm. This is the notochord. This is the paraxial mesoderm, intermediate mesoderm, lateral mesoderm, right? This is the amniotic cavity. And then this is the umbilical vesicle, right? And then remember, all around this is this great big balloon that they don't show in this view. I think they're going to show it in a minute, but there's this great big chorionic sac surrounding this whole thing, okay? Onward. So as you can see, the lateral side of the amnion begins to come down as the neural tube begins to close. The intraembryonic coelom gets bigger and everything pinches together at the end, or at the belly, at the middle. So now you've got the gut tube here. This is the neural tube. These are the neural crest right here. And then this amniotic sac now surrounds the entire embryo, right? And this is part of the umbilical cord now. Moving on. So this is going to be from the I guess transverse, just from a different, more cranial view, or a more cranial transverse section. Same idea though. This intraembryonic coelom becomes large. The embryo, or the amniotic sac, surrounds the entire embryo. Neural tube, neural crest, gut tube, etc. So now they're going to show us the sagittal plane. Oh, see, and here they show this is the chorionic sac. This is the big balloon of water that all of this lives inside. So that's actually a really good view of it. All right, so you're going to see all of this folding over. The heart's getting bigger as this happens. The brain is getting bigger as this happens. And all of this is going to get pinched together. I don't know why they didn't show it, but all of that, let's back that up just a tiny bit. All of this is eventually going to get pinched together with this to form the umbilical cord. And you notice how the amniotic sac, I like how they showed that in this view, it's already getting bigger, and it's eventually going to get so big that it fills this entire space. And so on and so forth. Right? So let's go over for a minute now. That's going to be it for embryonic folding. We're going to spend a minute on which tissues become which structures in the body. Some of this is definitely going to be on the quiz. So this chart, I forget which page in the book this is on. Let me look real quick. We're in chapter. Oops, I'm way past it. So this is on page 53 in the textbook, if you have the paperback, if you have the hard copy of the textbook. But yeah, so down here, you'll notice everything starts as embryoblast, okay? And all of the tissues of the embryo itself are ultimately derived from the embryoblast cells. These embryoblast cells at this stage of the game are considered to be omnipotential. Omnipotential cells, let me just turn this on so it'll stop turning off. Omnipotential cells can basically become any type of tissue in the human body. They can become any cell type. As soon as they differentiate into the different germ layers, they change from omnipotential cells into pluripotential cells. And then with each level of differentiation, as we go down through the process, with every level of differentiation, the variety of possible cell types decreases until finally a cell can only reproduce that same type of cell, or in a lot of cases, such as the brain and the muscle and things like that, they can no longer reproduce at all. They can't divide and make new cells once they differentiate past a certain point. Okay, and so you'll notice, you know, there's some trends here, right? You have you know, the mesoderm that again is going to become all of the supporting structures, right? It's going to become the cranium, the connective tissue of the head, the dentin, the muscles, the striated skeletal muscles, the limbs, the long bones, right? It's going to become the urogenital system. It's going to become the connective tissues of the viscera and the organs. The blood and the lymph cells and the spleen and the anyway, it's going to become all of the supporting type of structures rather. The ectoderm, right? The ectoderm is going to become all the surface structures. It's going to become the epidermis, the hair, the nails. It also becomes, interestingly enough, you know, the teeth, the inner ear, the lens of the eye, the anterior, but these are all skin type structures, right? Structures. And then you have, so that's the surface ectoderm. The neuroectoderm is going to become, you know, neural crest and neural tube and all the things that those guys turn into. Whereas the endoderm is going to become all the most internal structures. It's going to become the epithelium, the epithelial lining of the trachea, the bronchi, and the lungs. It's going to become the epithelial parts of all of the organs and all of the gut tube. It's going to become all the internal stuff, the epithelium of all the guts and organs and internal stuff. So there's like a pattern to what all these different things become. That's why they call them endoderm, because the endoderm becomes kind of the internal things, and the ectoderm becomes kind of the external things, and the mesoderm becomes the in-between things, all the supporting structures of the body. So you'll want to have kind of a basic knowledge of this for the quiz. I don't remember asking a whole lot about it, but you want to definitely have just a basic knowledge of which kind of structures come from which primordial tissues from this graph. Okay. Now, how does the body control, you know, how this decide which cells become these and which cells become those and which cells become, how does the body decide which cells become, or which progenitor cells become which types of tissues? So the answer to that is through this thing called inducers. So inducers can be a lot of different things. Sorry about the kids in the background. Inducers can be a lot of different things. Inducers can be diffusible chemicals or molecules. They can also be a matrix that's secreted by another cell type. They can be physical contact between two cell types, such as the notochord, right? The notochord physically contacts the ectoderm and stimulates neurulation through that physical contact. But ultimately, there has to be some kind of an inducer, and then the cell that's being induced has to have some type of a receptor or some other way to recognize that inducer. Okay, if one of these is missing, things go very, very, very wrong, and the embryo usually dies, which of course is either a miscarriage or a spontaneous abortion, depending on the stage of development you're at when this happens. If the embryo happens to survive that error, then it's going to have some kind of significant defect when it's born, right? So changes of one cell type to another are ultimately moderated by the DNA. It's the DNA that responds to these inducers and creates these change. So this is called epigenetics, right? The response of the DNA to these induction signals. So epigenetics controls the expression of genes. Everybody has the same genes, right? I have the same DNA as, you know, my Polynesian buddy who has much darker skin and dark, curly, tight, you know, we look a lot different, but we have the same genetic code. So how does that happen? Well, that's through epigenetics, because the epigenetics controls the expression of the genes. Me and him have the same number of melanocytes in our skin, but his skin's a lot darker than mine because his melanin genes express that trait and mine do not. Bummer for me. I have to sunburn when I go out in the sun, or else I have to cover my skin or wear sunblock, and he doesn't. That's just how it is. Yeah, so epigenetics right now is an exploding research field. They're understanding more and more every day. In fact, the first time I taught this class, they understood an awful lot less than they do now. It's a hugely exploding field. We still have a long way to go, though. We have a lot to learn about what causes the expression of some genes or inhibits the expression of some genes. And, you know, it's really cool to me that we still have so much to learn about the body. I just think that's amazing. I always get a kick out of, you know, the anatomists that have the PhD and think they know everything there is to know about the body, or the physiologists. Man, that is a load of baloney. We have so much still to learn. And the amount that we still have to learn is amazing to me. I look forward to some future day, if I qualify, to be exalted and become, you know, like God and have this omniscience where I just know everything. Man, I can't wait to find out how some of this stuff works. But for now, I have to just learn a little piece at a time with my very minimally capable human brain in this mortal body that is so incredibly amazing. At any rate, that's going to be the end of this lecture. I hope I gave you a few things to chew on. I personally think that this embryonic folding process is really, really, really amazing. In fact, let me back up the slides a little ways. So when we're at this stage, when we're still at an embryonic disc and we have all this neural plate stuff going on and neural groove, and we haven't even fully formed the neural tube yet, innervation has already begun. So the nerves are already going out right here. And then after the nerves begin going out, then you have this process of the embryo folding laterally and folding ventrally. And so those nerves follow the tissues that they're innervating as they move around the body. And this is one of the things that causes, you know, referral pain. That's why you can poke somebody in the shoulder and it hurts in their wrist, or you can poke them in the middle of the back and it hurts in their head. And all these things that seem so strange largely happen, or at least in part happen, because of this folding of the embryo. And because the nerves happen to start going out at this stage of the game, and then they follow the tissues as this thing folds and changes shapes because they have to. Again, sorry about all the noise in the background. I did this lecture at my house on a Saturday, and the kids are being crazy noisy today. Maybe you enjoyed that. In any case, that's going to do it for lecture number 7. And we'll see you at lecture number 8. Aloha.